My Life HIV edition. I was first discovered among men who have sex with men. But later on, a researcher known as Robert Gallo, whose main research was with the Institute and focused on T cell leukocytes, which is a subset of white blood cells which are key to the body's immune response. The foundational research led Gallo and his team to grow T cells and isolate viruses that affect them, including one called the human T cell leukemia virus, or HTLV. When the news of a mysterious gay cancer were first reported in the US in 1982, Gallo and his team turned their attention to identifying what they believed to be a viral agent, causing the rapid depletion of T cells in sick and dying patients. Gallo and his team isolated a virus they labeled HTLV3, after which it was renamed HIV in the laboratory. Robert Gallo discovered that there were two types of HIV, in which I was one of them, HIV-1 and HIV-2. HIV-1 was originated from pan troglodytes or chimpanzees in the Western Africa, which spread to humans when they were hunting for bush meat, which is otherwise known as meat of wild African animals.
HIV-1 and HIV-2 both resulted in AIDS. However, HIV-1 was known as the more lethal virus as it caused a lower CD4 count and persisted in the human body for longer periods of time. Now, a little bit more information about me. I'm a retrovirus consisting of 3 separate layers.
I may look like a planet, but trust me, I'm the last planet you would ever want to live on. Can you imagine a planet that will slowly kill you from the inside without you even knowing that's happening?
My outermost layer is the lipid envelope, followed by the caspase protein, and the innermost layer housed my single-stranded RNA. Ignore what this incompetent human wrote on the board, as he obviously doesn't know the difference between a single-stranded RNA and a double-stranded RNA. Furthermore, those 3 looking things you see in that diagram, well, those are my membrane proteins that are used to interact with the CD4 cells of T helper cells. The genome that I hold codes for 3 important proteins that are used in my attack on T helper cells.
The first of which is TET, which is used to activate my viral transcription. Next, REV, which is responsible for the transport and stability of the viral RNA that I house. And finally, NEF, which is active in downregulations of CD4 cells belonging to the T helper cells which I have attacked.
My attack on the T helper cells are rather simple. I first attach to the CD4 receptor on the surface of T helper cells using my membrane proteins, and I also interact with the CCR5 co-receptor on the same T helper cell, which then allows me to insert and transfer my viral genome into the T helper cell. My viral genome then enters the cytoplasm into the nucleus to undergo transcription. Following that, my viral genome is then transferred into a place near the surface membrane of the T helper cell and is budded off, forming new copies of me to continue my assault on the human body.
Once I have attacked the T helper cells, they are no longer able to activate the immune response of the human body.
T helper cells, which secrete cytokines, can no longer activate the B cells, the cytotoxic T cells, and also other T helper cells. The B cells are responsible for proliferating into plasma cells and also memory B cells. Cytotoxic T cells are involved in secretion of toxic chemicals, and finally, the other T helper cells continue to activate the immune response, which will halt the assault of other pathogens which aren't me. These cytokines released by T helper cells are also used to attract macrophages, which will attack non-self antigens that have entered the human body. Thus, they would then engulf the non-self antigen and present the antigens, attracting more T helper cells to the site of infection. And if you think my attack just stops there, you are wrong. I continue to assault the human body by attacking even more T helper cells by proliferating from one to many of copies of me. This stops the T helper cells from carrying out their function, essentially weakening the immune system of humans. Thus, they are more susceptible to weaker pathogens and are more easily infected by diseases such as influenza and chronic diarrhea. These diseases, which are usually not fatal, would end up killing this weaker form of the humans due to AIDS, which I am the main factor of.
AIDS stands for Acquired Immunodeficiency Syndrome, which is the leading cause of death. CD4 counts is affected when someone is affected by AIDS. CD4 count will decrease dramatically from around 1,500 to 500 when someone is infected with AIDS. The picture below is just an exaggeration of the CD4 count. So symptoms of AIDS include chronic diarrhea, severe weight loss, high fever, and ultimately it causes death. Can be transmitted in various ways, mostly through bodily fluid. Bodily fluid includes semen, which is ejaculated from the penis, and also vaginal fluid from the vagina, also blood transfusion, and breast milk from the mother given to the baby. Normally, bodily fluid will not be transmitted from one person to another, but it can happen in several methods. One of them being sharing of needles between infected and non-infected person. Also, it can be transmitted through unprotected sexual intercourse. And bodily fluid can also be transmitted to the fetus from an infected mother through the placenta. Blood transfusion from an infected person can also cause the person receiving the blood to acquire AIDS. Now, a little explanation on why HIV can only be transmitted through bodily fluid and not other blood-sucking animals such as the blood-sucking bat and mosquitoes, and also insects such as leeches. In order for HIV to be transmitted into a person, the HIV must enter the bloodstream. Thus, only bodily fluid are able to do so, as they can pass through the mucous membrane. So far, there is no cure for AIDS. This is because HIV has antigenic concealment property. This allows it to hide inside the host cell, and it cannot be detected by the antibody of the human body. This allows HIV to stay in the body for as long as it wants. Unfortunately, my friends and I can be prevented if human does regular hospital checkups, practice safe sexual intercourse by using condom. Also, used needles should not be reused. It should be disposed each time after every use. Humans should also avoid premarital sex, as this would increase the chance of having AIDS. Next up, breastfeeding is also a cause of HIV. Thus, mothers should use formulated milk such as Dugo to substitute for breast milk. Here comes the people that I hate the most, because they are resistant to me and my friends. This is due to a mutation in their gene that causes their CCR5 receptor to be absent, and thus prevents me from entering into their cell. This is known as the CCR5 delta 32 mutation. So, what's the difference between a HIV resistant person and a non-HIV resistant person? Is that a non-HIV resistant person has the CCR5 co-receptor, which I can bind to and help me to enter the human T helper cell. In contrast, with the HIV resistant person, their CCR5 co-receptor is absent. Thus, I will not be able to interact with it and release my viral genome, and subsequently unable to initiate any attack on the human. Thus, a non-HIV resistant person is vulnerable to AIDS, whereas a HIV resistant person can live worry-free of AIDS. Lastly, here is a message from a human friend of yours.
We hope that you have learned and absorbed all the knowledge that we have provided, and if you have any inquiries or questions, please do not hesitate to email the email provided. We will try to get back to you with answers as soon as possible. This is Harnish Sinkara, and on behalf of Daryl Ng, Kenneth Law, Ian Law, and of Group 1, we would like to thank you for watching our video. We wish you all the best in the upcoming exams, and always remember, my friends, stay safe, stay happy.